European rabbits Eryctolagus cuniculus were initially brought to Australia in 1788 by the First Fleet, but it was Thomas Austin, an English settler, who is most famously associated with their widespread release in 1859. Although rabbits had been introduced earlier, Austin's actions significantly contributed to their rapid proliferation across the country. These wild rabbits have since become a major invasive species, causing extensive damage to crops and resulting in economic losses worth millions. Their rapid spread was likely aided by the development of resilient crossbreeds. Throughout the 20th century, numerous efforts were made to control the rabbit population in Australia. Traditional methods, such as shooting and destroying burrows, met with limited success. One major initiative, the construction of a rabbit-proof fence in Western Australia from 1901 to 1907, ultimately failed to stop their spread. A more effective measure came in the 1950s with the introduction of the myxoma virus, which caused a dramatic decline in rabbit numbers. However, the population eventually began to recover as some rabbits developed resistance. In response to their invasive nature, Queensland has prohibited keeping domesticated rabbits as pets. Rabbits were first brought to Australia in 1788 by the First Fleet. This fleet was made up of 11 British ships that carried settlers, including over 1,400 convicts, soldiers, sailors, and officials, to start a new British colony. Led by Captain Arthur Phillip, the fleet left Portsmouth on May 13, 1787, and arrived at Botany Bay on January 18, 1788, after a journey of over 24,000 kilometers. However, Phillip chose Port Jackson, just north of Botany Bay, as the site for the new colony. They arrived there on January 26, 1788, and this marked the founding of New South Wales as a penal colony, the first British settlement in Australia. The rabbits were likely kept in cages and used as a food source in the early years, and they were not very common, as shown by their absence in early archaeological food remains. However, by 1827, rabbits had become very numerous in Tasmania. A newspaper at the time reported that thousands of rabbits were roaming freely on large estates. In contrast, New South Wales still had no wild rabbit populations. Explorer Cunningham mentioned that rabbits were only kept near houses and not yet living wild, although he believed the scrubby, sandy land between Sydney and Botany Bay would be perfect for raising them. This shows that while Tasmania was experiencing a rabbit population boom in the early 1800s, New South Wales was not. In early colonial Australia, enclosures likely referred to larger, walled rabbit farming areas rather than small cages. One of the first known examples in Sydney was built by Alexander Macleay at Elizabeth Bay House. He created a stone-walled rabbit preserve that was well stocked for game hunting. By the 1840s, keeping rabbits became more common, with court cases reporting rabbit theft from homes and rabbits becoming a regular part of people's diets. In 1857 to 1858, Alexander Buchanan, the manager of F. H. Dutton's Allaby estate in South Australia, released rabbits for hunting. Their numbers stayed relatively stable until about 1866, likely because native predators were keeping them in check, and rabbits were even protected by law at the time. However, by 1867, their population had grown out of control. This rapid increase was initially blamed on the loss of native predators, but later studies suggested that a stronger, more adaptable rabbit breed had emerged through natural selection, helping them spread even further. The current rabbit infestation in Australia is believed to have started in October 1859, when Thomas Austin released 24 wild rabbits on his property, Barwon Park, near Winchelsea, Victoria, for hunting. By 1866, the Geelong Advertiser reported that hunters had already killed around 50,000 rabbits in the area. Austin, originally from England, had been a keen hunter and enjoyed rabbit shooting on weekends. After moving to Australia, where rabbits were not native, he wanted to recreate his hunting experience. He asked his nephew William in England to send him 12 grey rabbits, 5 hares, 72 partridges, and some sparrows. He believed that releasing a few rabbits would do no harm, and would bring a bit of England to his new home. Because William couldn't find enough wild grey rabbits, he supplemented the order with domestic ones. One theory suggests that the rabbits at Barwon Park thrive so well because the crossbreeding of wild and domestic rabbits produced a hardier hybrid better suited to the Australian environment. Inspired by Austin, other landowners also released rabbits into the wild, worsening the spread. Rabbits multiplied rapidly and spread quickly throughout the southern parts of Australia. The country's mild winters allowed them to breed all year round, and farming practices transformed natural scrubland and woodlands into open areas with low vegetation perfect for rabbits to thrive. Rabbits are most comfortable in areas with a mean annual temperature above 10 C and a mean warmest monthly temperature of 25 C or lower. They get most of their water from the plants they eat, except during droughts when they need access to water within about 0.4 kilometers of their burrows. Rabbit populations tend to be highest in areas that receive more than 400 millimeters of rainfall annually and have winter temperatures above 4 C. As these conditions promote plant growth, rabbits prefer burrows in soils with 20-50 clay content or in deep, sandy soils. Additionally, the presence of predators, diseases including biocontrols, and control efforts by land managers such as baiting, warren ripping, and fumigation all affect rabbit populations at local or regional levels. This led to one of the most extreme cases of unintended consequences. Within just 10 years of their release in 1859, rabbits had become so numerous that around 2 million could be hunted or trapped each year without making a dent in their population. It became the fastest spread of any mammal ever recorded. Today, rabbits are firmly established across southern and central Australia, with some smaller populations found even in the northern deserts. Although rabbits became a serious pest in Australia, they were also useful to many people during tough times, like the economic depressions of the 1890s and 1932, and during wartime. Trapping rabbits gave farmers, stock workers, and station hands a source of food and a way to earn extra money, sometimes even helping them pay off debts. Rabbits were fed to working dogs, boiled for poultry feed, and later sold frozen for local use and export. Their skins were valuable too, used in the fur trade and still in demand for making felt hats. However, since their arrival from Europe in the 19th century, rabbits have caused major ecological damage in Australia. They are believed to be a leading cause of native species loss. Their overgrazing has severely reduced natural plant cover, and when grasses run out, they feed on shrubs and even the bark and leaves of trees. This often leads to the death of young trees, especially in orchards and forests, though the full extent of the damage to plant species is still unknown. Rabbits have also caused major erosion issues in Australia. By eating native plants, they leave the soil bare and unprotected, making it more likely to be eroded by wind, rain, and water runoff. This erosion strips away the topsoil, which is vital for plant growth, and can take hundreds of years to naturally recover, making the damage long-lasting and severe. By 1887, the damage caused by rabbits was so bad that the New South Wales government offered a £25,000 reward, around $3.9 million in today's money, for a new and effective way to completely get rid of them. Over 1,400 ideas were submitted, including some that suggested using biological controls, but none of the proposals were both safe and effective enough to be used. In 1901, a royal commission was held to investigate the growing rabbit problem in Australia. Once the scale of the issue was fully understood, several methods were introduced to try to control or reduce rabbit numbers. However, these early methods had limited success until biological controls were introduced in the second half of the 20th century. One common method is shooting, which helps manage small rabbit populations and can also provide food for people or pets. However, it's not practical for wiping out large numbers. Another widespread method is destroying rabbit warrens. This is done by dragging sharp tools through the ground with bulldozers, which can dismember or bury rabbits alive, plowing, blasting, or fumigating. On large farms called stations, These techniques are frequently used. Australia's sandy soil makes ripping and plowing especially effective, and both tractors and bulldozers are used in these operations. Poisoning is one of the most commonly used traditional methods to control rabbit populations in Australia. It requires minimal effort and can wipe out rabbits in a local area. But because rabbits are highly mobile, reinfestation often occurs. One of the earliest poisoning techniques involved using bait made of pollard, a grain byproduct mixed with a phosphorus-based poison, like sapapor from the company Sayers, Allport, and Potter. Phosphorus has the advantage of breaking down into harmless phosphoric acid in dry weather, making it safer for livestock and pets if used correctly. However, it's also flammable and can release harmful fumes. Modern poisons used for rabbit control include sodium fluoroacetate, known as pindoni. Another method is hunting with ferrets. Ferrets are released into burrows to flush rabbits out, so they can be caught in nets or shot. While this is more of a traditional or recreational hunting method rather than an effective population control strategy, it has been used. Unlike in Europe or the US, Australia lacks many natural mustelid predators like ferrets or weasels, which limits natural predation on rabbits in their burrows. In the past, trapping was commonly used to control rabbits, but steel-jawed leg-holding traps were banned in most states in the 1980s due to animal cruelty concerns. Today, rubber-jawed traps are still used, but trapping is less common and mainly done in settled areas. These methods require a lot of work. Ring fencing, which involves enclosing an area with a fence to keep rabbits out, can be very effective. In the 1880s, James Moseley used wire netting to fence Coondambo Station, and the rabbits died of thirst during a heat wave. Later, he used similar fencing on other stations in the Gawler Ranges, transforming degraded land overrun by rabbits into profitable sheep farms. Modern examples of successful ring fencing include the Warrawong and Yucamora wildlife sanctuaries, which were created by John Walmsley. These sanctuaries also keep out other animals like foxes, dogs, and cats. In July 1884, Ernest James Stevens, a member of the Queensland Legislative Assembly, suggested that the Queensland government build a fence to stop rabbits from spreading into Queensland from New South Wales. In 1893, work began on the rabbit-proof fence in Queensland, which was extended over the years. The final section was completed in 1997, connecting it to the dingo fence. It stretches from Mount Gyps near Rathdowney to Goombie, between Chinchilla and Miles. From 1901 to 1907, a similar rabbit-proof fence was built in Western Australia between Cape Caraudran and Esperance to stop rabbits from spreading eastward into Western Australian grazing areas. Since European rabbits can jump high and dig burrows, a long, unbroken fence was unlikely to succeed. The first rabbit-proof fence, built in 1901, failed to keep rabbits out. Despite this, smaller-scale fencing projects later proved successful in controlling the rabbit population. Releasing diseases that affect rabbits has been somewhat successful in controlling the rabbit population in Australia, though not as quickly as expected. In response to the New South Wales government's offer of a £25,000 reward for a biological control method, Louis Pasteur became involved and suggested using viral methods, such as the chicken cholera bacillus now known as Pasteurella multicida. Although this approach didn't work, Pasteur's involvement helped introduce microbiology to Australia. On April 16, 1888, the New South Wales government established a royal commission to investigate whether introducing contagious diseases to rabbits could harm human health, other animals, or agricultural activities. The commission conducted tests on Rhode Island, where rabbits given food with chicken cholera bacillus died, but no evidence showed that the disease spread to healthy rabbits. In 1885, Professor Archibald Watson from the University of Adelaide suggested releasing rabbits infected with rabbit scab into an enclosed trial area. However, early tests showed that this method would likely be ineffective in Australia's drier regions. In September 1887, Dr. Herbert Butcher from Wilcannia discovered several dead, emaciated rabbits at Tintinology Station. He and Dr. H. Ellis from Sydney believed the rabbits had died from a new disease, which they named the Tintinology virus. They thought it could be a potential control method, but the disease was never confirmed to be contagious or infectious. It might have simply been caused by starvation due to harsh natural conditions. In 1906 and 1907, Jean Danish from the Pasteur Institute in Paris tested a strain of Pasteurella bacteria on Broughton Island, New South Wales. This strain was specific to rabbits, but it didn't provide an effective solution. Danish believed the island was a poor location for testing and suggested that further experiments should be done on the mainland. After Danish left in 1907, his Australian collaborator Frank Tidswell continued the trials, including experiments with microbes from the Yalgagrin, Gundagai, and Picton stations, where infected rabbits had been found. However, the trials lacked financial support from the federal government and the cooperation of affected states, preventing the necessary evidence to prove the methods were both safe and effective. The fact that these diseases didn't eliminate entire rabbit populations may have caused doubts about their effectiveness. In 1950, Frank Fenner's research led to the deliberate release of the myxoma virus into the rabbit population, which caused their numbers to decrease from around 600 million to 100 million. However, over time, some rabbits developed genetic resistance, allowing their population to recover to 200 to 300 million by 1991. To address this recovery, from June 1991, the Commonwealth Scientific and Industrial Research Organization CSIRO tested the potential of a calicivirus, which causes rabbit hemorrhagic disease RHD, for biological control. The virus accidentally escaped from a quarantine area on Wardang Island, South Australia, and by late October 1995, it had spread to areas in northeastern South Australia. By the winter of 1996, it had reached Victoria, New South Wales, the Northern Territory, and Western Australia, detected by analyzing the livers of dead rabbits. The virus was more effective in dry areas, while in colder, wetter regions, a less harmful calicivirus was found to be giving rabbits some immunity. In Australia, there is a legal vaccine for RHD, but no cure is known for either myxomatosis or RHD, and affected pet rabbits often have to be euthanized. In Europe, where rabbits are farmed widely, they are protected from both myxomatosis and calicivirus, with the genetically modified virus developed in Spain. In 2012, virologist Francisco Parra and his team at the University of Oviedo in Spain discovered a new variant of the rabbit hemorrhagic disease virus, a highly lethal and contagious strain known as K5RHDV. In 2017, Australian authorities released this strain at around 600 points across the country. As a result, domestic rabbit owners were advised to vaccinate their animals. In southern Europe, the decline in rabbit populations is a concern for the conservation of endangered predators, like the Iberian lynx and the Iberian imperial eagle, which rely on rabbits as prey. This situation highlights the potential impact of biological control methods used in Australia on wildlife conservation in other regions. By 1920, Australia had an estimated 10 billion rabbits. Today, the population is about 200 million, and they occupy 70% of the country's land area, 3 million km. In Queensland, rabbit populations have varied significantly over the years, 150 million in 1949, before the introduction of myxomatosis, 5 million in 1995, before the release of RHDV, 2 million in 1996, shortly after and 14 million in 2008. Rabbit numbers can fluctuate greatly depending on the seasons and the effectiveness of biological controls. Their rapid breeding means they can quickly rebound after droughts or the introduction of new biocontrols, though this recovery is slower after a new biocontrol. as rabbits develop immunity over time. In conclusion, rabbits in Australia have become a major environmental and agricultural concern due to their rapid population growth and destructive behavior. Initially introduced in the 19th century, their numbers exploded to an estimated 10 billion by 1920, with the population currently around 200 million. Environmental factors like temperature, rainfall, and soil type influence their distribution, while the availability of food and water also plays a crucial role. Despite various control measures, including shooting, trapping, fencing, and the introduction of biological controls like myxomatosis and rabbit hemorrhagic disease, rabbits remain a persistent challenge. These methods have had varying levels of success, but their populations continue to fluctuate, often recovering quickly after droughts or the introduction of new control measures. As a result, ongoing efforts to manage their impact on Australia's ecosystems and agriculture are essential.